In this video for Lecture 30, we'll cover the training adaptations that mostly affect endurance performance. Keep in mind, when we're talking about endurance performance, we really are talking about an effort that is, of course, prolonged, 30 minutes, 60 minutes, 2 hours, and is necessarily then done at an exercise intensity that is submaximal, and will mostly be done at what we could consider a steady state. And to do that, to maintain a steady state over prolonged periods, will require homeostasis, or the maintenance of homeostasis, at the same time as you're trying to maintain the highest power output or the fastest running pace or swimming pace that you can do. It turns out most of these adaptations that we're going to talk about occur within the muscle itself. These are biochemical changes and not as much to do with the cardiovascular adaptations we saw in the previous lecture. So first, let's take a look at the outline. And the main heading for this section for this set of pages is Endurance Training, Effects on Performance, and Homeostasis. And the page numbers for this are going to be 299 to 305. But the subsections in this main section then are going to be fiber type and capillarity, mitochondrial content, plasma glucose concentration, muscle antioxidant capacity, which we are going to skip so we can cross that one out. And last we'll end up with acid-base balance. We will see as we go down the list that almost all of these are really going to be related to the increased mitochondrial content in the muscle fibers. Let's first take a look at the changes in fiber type and capillarity that occur. And so first, talking about muscle fiber types, what we see with endurance training is that there is a shift from fast myosin isoforms to slow myosin isoforms. So that is, the percentage of your slow myosin will increase with endurance training. The absolute magnitude of this shift will depend on how much training is actually done, the intensity of the training, and of course the duration. And we also know this can occur over years. So analysis of endurance athletes who have been doing this for a long time will show that even after many years of training, they still will be slowly and progressively increasing their percentage of slow twitch fibers. However, that being said, the primary determining factor for your percentage of slow twitch fibers is probably genetics. It's what you're born with. So with training, we might see a 5 or 10 or maybe at most over many, many years, a 15% shift in 15% increase in the percentage of slow twitch fibers. The ultimate effect of this, of course, is that slow twitch fibers are more efficient than fast twitch fibers, and so having more of them would allow you to be more efficient over long-term endurance performance, as well as, keep in mind, recruitment of slow twitch fibers happens first, and so the more slow twitch fibers you have, the more you can recruit, which means also if you're thinking about recruitment, you can produce more power with those slow twitch fibers, and so you can then run at a faster pace or cycle at a faster speed and still be only recruiting slow twitch fibers. So there is an advantage. The second factor for this section is the increase in capillary density. And so again, with this, we would see more capillaries per area, or sometimes you'll even see it described as more capillaries per fiber. And so what it does is, the major issue is that there is a shorter transit distance or diffusion distance for oxygen to get from the capillary to the mitochondria. And so this will enhance the rate at which oxygen can be used, so therefore it'll enhance your VO2 max. But it'll also enhance the exchange of wastes, and so the removal of metabolic waste can also be done more readily. And so, of course, homeostasis can be better maintained, and the person will then be able to maintain a higher power output or running or cycling pace. The next section covers the increase in mitochondrial content that occurs with endurance training. Now, this is one of the hallmark responses of endurance training. That is the increased mitochondrial capacity. And as we'll see, it is this adaptation that probably has the biggest effect on performance because the increased mitochondrial capacity is what helps to maintain homeostasis in the different ways that we'll see going forward. But for right now, let's look what it does metabolically right at the outset. And so we'll look at figure 13.3, which shows us what happens when mitochondria double in size. We tend to think of the adaptation being we get more mitochondria. Actually, now we know mitochondria actually just get bigger, so they increase their total volume and take up a greater portion of the muscle fiber. And so if we have at the top of this figure, we can see that a single mitochondria here, mitochondrion, if we look here, this 100 units of ADP, that would represent our breakdown of ATP into ADP. And so our rate of cross-bridge cycling, let's say. So our energy utilization. That then has to be transported to the mitochondria, across the mitochondrial membrane, and then rephosphorylated to make ADP. And in that process, let's say we have a VO2 of 2 liters a minute. That might be the steady state exercise VO2 that's required to maintain this, say, 15 miles per hour on your bicycle. The importance of this here shows that, of course, ADP is necessary because it's one of the substrates for oxidative phosphorylation. The other one, of course, is inorganic phosphate. And so as ADP arrives at the mitochondria, then that will stimulate the reactions that then require the oxygen uptake. So now let's look at the situation after training. So we have a larger mitochondria, and that means it has more surface area. It can transport ADP and ATP across its membrane more readily because there's more surface area. And notice that the intermitochondrial membranes are also of greater volume. That means they are full of more enzymes, so Krebs cycle and electron transport chain, and those then now have an increased capacity. The point of this lower figure now is that with, let's say, double the surface area, now we can get it, or the mitochondria can import just as much ADP, let's say 50 units at one portion and 50 units at another portion, to generate this 2 liters per minute oxygen uptake. But notice that it can be done at a smaller ADP concentration. Essentially what this is saying is that now the mitochondria are more sensitive to ADP. That is, they can pump out ATP at a given rate that requires, say, 2 liters a minute oxygen uptake, but they can do it at a smaller ADP value, which means there is less challenge to homeostasis, right? The change in cellular ADP is now less. I'll try to illustrate this in another way because I know this is a difficult concept. I've taken a previous figure in your book that actually, instead of increasing the size, they increase the number, and I think this one sort of makes sense. So if we look at the top, 100 units of ADP going in gives us a VO2 of 2 liters a minute. That's just like the one we saw before. But now, so instead of doubling the size of the mitochondria, the previous version of your book just made 2 mitochondria, and they said, what if we get more mitochondria? Well, now each one of these can have an uptake of 50 units of ADP, which now it's cut in half, right? And only need 1 liter per minute, which then, of course, yields 2 liters per minute, which is the same VO2 as what happened above before training. But now with training, of course, again, we can accomplish the same oxygen uptake with a smaller concentration of ADP. A final and third way to look at this, as I created this figure on the right side, and going back to the top, before training, if we have 100 units of ADP in the cytoplasm, that will generate an oxygen uptake of 2 liters a minute. After training now, if each of these stayed the same, this 100 units of ADP, so now I've just doubled it from 50 because I've kept the same input to the mitochondria, that of course each one would still give us a VO2 of 2 liters a minute. But now since we have double the mitochondria, our overall VO2 could be 4 liters a minute. And so what I did there is I've just showed us how we can increase our capacity if we have more mitochondria. I don't want to get too down into the weeds on this and all of these details. And again, I'll go back to the main point of this, which is with more mitochondria, we can maintain homeostasis better, that is, the metabolites within the cytoplasm, because a smaller stimulus is needed to to produce the same VO2. For example, 50 units of ADP rather than 100. And so the challenge to homeostasis has now been reduced. We can more easily handle this workload of, say, 15 miles per hour. The final point for this section can be made in looking at figure 13.4, and this shows us, if we look at the x-axis, that's time, minutes, and this would be over the case of the beginning of a submaximal exercise bout where we've reached a steady state. And then on the y-axis is your oxygen uptake. Note that in the trained state, after training is the green, there is a faster rise in VO2, and the person reaches a steady state sooner than before training. So in this case, steady state is reached at 2 minutes, whereas in the before training, maybe at about 3 minutes. So how does this improve performance? Well, if we look at the little pink box here, the effect is, of course, you get this faster rise in oxygen uptake, and then because of that, you rely less on glycolysis. Why? Because now we're relying more on oxidative phosphorylation, right? You have a lower oxygen deficit. And when there's less reliance on glycolysis, you'll have less lactate formation. And if there's less reliance on creatine kinase reaction, you'll have less phosphocreatine depletion. And so your overall homeostasis is improved. As we now move into the next section, we'll see another important feature of an increased mitochondrial content, and that is a maintenance of plasma glucose. And I'll use figure 13.6 here to explain the finer points of this. First off, we have the end point. Notice on this figure is a sparing of plasma glucose, and that is because we have an increased use of free fatty acids. And the three main mechanisms for this, I've listed underneath here the main point, which is maintenance of plasma glucose. You have an increase in the transport of free fatty acids into the muscle, a transport of free fatty acids into the mitochondria, and then a greater oxidation of free fatty acids in the mitochondria. So let's take those three mechanisms one at a time. We'll start at the far left of this figure or this diagram and see that, of course, our stimulus here is going to be endurance exercise training. And let's go up to the right, this increased capillary density. Keep in mind we're heading towards an increased use of free fatty acids or enhanced oxidation of free fatty acids. And I should point out, too, this is one of the other hallmark adaptations of exercise training, is that a trained person and trained muscle fibers then are better at using free fatty acids as fuel than the non-trained muscle. And so a higher percentage of ATP will be formed from breakdown of fatty acids in a trained muscle than it will be in an untrained muscle. The consequence, of course, if you think about what we've done with metabolism earlier in the semester, the RER will be lower because the person will be burning a relatively greater percentage of fats. That would be at any given workload. So back to the diagram, we can see an increase in capillary density. This has the effect of slowing down the blood flow and creating a greater surface area for the exchange of free fatty acids. The other adaptation we'll see in skeletal muscle is an increase in the transporters for free fatty acids to transport them from extracellular fluid into the intercellular cytoplasm. And so these are proteins that will be embedded into the plasma membrane, the sarcolemma, and there are two of them. One of them is called fatty acid binding protein, FABP, and the other one is so appropriately named FAT, FAT, which is fatty acid translocase. So both of those transporters will increase in the sarcolemma of trained muscle fibers and therefore increase the transport of free fatty acids into the muscle. Our second step for increasing free fatty acid utilization will be the transport of free fatty acids into the mitochondria itself. So for example, the first mechanism here, now we'll go down on this diagram. With more mitochondria, or larger mitochondria, we have a greater mitochondrial surface area, which means we can accomplish the import of more fatty acids into the mitochondria. In addition, go to this bottom box, we also see an increase in the carnitine transferase. I'll explain this. This is one of the enzymes that transports free fatty acid across the mitochondrial membrane. Indeed, there are two of these. The first one is called carnitine palmitoyl transferase 1, or CPT1, and the other, also called FAT. This is mitochondrial fatty acid translocase. Both of those proteins will have increased concentration in the mitochondrial membrane, which then also, again, will now enhance the ability of mitochondria to take in free fatty acids for oxidation. The third mechanism for maintaining plasma glucose is a mitochondrial oxidation of free fatty acids. And so in this case, we have an increased ability. Obviously, now the mitochondria can import more free fatty acids. Now we have the increased ability to oxidize those fatty acids. And that's shown in this square down here at the bottom. Increased beta oxidation enzymes. So remember, beta oxidation is the breakdown of free fatty acids into acetyl-CoA. And so by providing more acetyl-CoA to Krebs cycle, we're going to get more ATP, obviously. So looking back over these three mechanisms, we have our increased transport that will be essentially manifest right here as an increased uptake of free fatty acids. We have increased transport of free fatty acids into the mitochondria, so this carnitine palmitoyl transferase 1 and mitochondrial FAT, increased beta oxidation. All of those yield then to an increase in free fatty acid utilization, which of course now will spare plasma glucose because we're using less glucose to power muscle contractions. The advantage here is that, of course, now the brain, which only uses glucose, has a more constant supply of its energy source. Now let's jump to the last section of this lecture. Remember, for Section D, this endurance training improves muscle antioxidant capacity. We're going to skip that. And so we'll go right to E, improved acid-base balance during exercise. Just like the last few topics, the increase in mitochondrial content in the exercised muscle, the trained muscle, is going to be paramount here in carrying out this effect. There are three main mechanisms to increase acid-base balance, and that is an increase in free fatty acid oxidation, increased mitochondrial uptake of pyruvate and NADH, and then also the transformation in the LDH isoform, that's lactate dehydrogenase. So that's one here that doesn't really rely on mitochondria. But to begin, let's talk about the formation of lactate, because we'll keep coming back to this reaction. So here's the lactate dehydrogenase reaction. Notice that pyruvate and NADH are transformed into lactate plus NAD. And so what the lactate dehydrogenase enzyme does is move the H from NADH and puts it on pyruvate to make lactate. So obviously from this, we know, well, from our previous lectures, that pyruvate and NADH are formed in glycolysis. And of course, this reaction shows one of their possible fates, which is possible fates of pyruvate, that is the formation of lactate. Pyruvate and NADH, of course, we also know can be transported and imported into the mitochondria for further oxidation in Krebs cycle and electron transport chain, where we're going to get the bulk of our ATP. So keep that in mind about this formation of lactate as we go forward, because it's important for the acid-base balance. We'll use the last figure in this section to discuss these three mechanisms for improved acid-base balance. The first one is an increase in free fatty acid oxidation. That is shown here in this square, increased free fatty acid oxidation and decreased PFK activity. That is a feedback mechanism. Remember, PFK is the rate-limiting enzyme for glycolysis, and the feedback molecule here is citrate, which of course is the first product of the Krebs cycle. So citrate feeds back and actually inhibits phosphofructokinase. So if we have lots of acetyl-CoA being formed from free fatty acid oxidation, we will then be going through Krebs cycle, have lots of citrate available that can feed back and inhibit glycolysis. When we do that, we get, of course, a decrease in pyruvate formation. And when that happens, of course, we're going to get a decreased lactate formation simply because now we've removed the substrate for the LDH reaction. Moving to our second mechanism, this would be an increased uptake of pyruvate and NADH by the mitochondria, and that's the lower square here. And this should make sense because, of course, if there are now a greater volume of mitochondria, we would have a greater capability of oxidizing pyruvate and NADH that are formed in glycolysis. If we go back to the previous figure in our LDH reaction, we can see, of course, pyruvate and NADH. One of their possible destinations is the mitochondria. If those go to the mitochondria, then, of course, we won't be able to make lactate from that pyruvate. Instead, the pyruvate will get modified into acetyl-CoA, which will then go to the Krebs cycle. The final mechanism for improved blood pH, well, and I guess interior muscle fiber pH, has to do with the isoform of lactate dehydrogenase. An isoform is just one of the forms of the enzymes. We call it an isozyme. It turns out lactate dehydrogenase is a tetramer. That is, it has 4 subunits, and each of those subunits is designated as either M or H. M stands for muscle, H stands for heart. And so there's a combination of those subunits that you can have, which makes 5 total different isoforms of the enzyme. The ones that are more M, say M4, are more common in fast-twitch muscle fibers. They have a high affinity for pyruvate. They like to take pyruvate and turn it into lactate. On the other end of the spectrum, the H isoform, and so those enzymes that are primarily H4, that would be more like what's in the heart, they have a low propensity, or low affinity, for pyruvate. They actually have a higher affinity for lactate. So they like to take lactate plus NAD and go backwards through that reaction and make more pyruvate that they can then use for oxidation. So with exercise training, we get an increase in the H4 isoform, which means it will just simply form less lactate. Its activity is less, and pyruvate will stick around longer. So all of these adaptations, that is, increasing your free fatty acid oxidation, increasing your pyruvate NADH oxidation, and increasing your H4 form of LDH, all have the effect of decreasing your lactate and creating H+, which then, of course, will decrease your pH. So if we reduce all those, we will be able to maintain pH at a higher level. So that wraps up the content for Lecture 30. As you can see, an increase in mitochondrial capacity, mitochondrial volume, as well as capillarity that occurs in the trained muscle create mechanisms then that allow for the trained cyclist or runner or swimmer to maintain a faster pace than the untrained athlete. And these are going to relate to the maintenance of plasma glucose, the increase in free fatty acid oxidation, better acid-base balance, and all of those things will just enhance the maintenance of an internal stable environment inside the muscle fiber, and so because, again, we have less of a challenge for homeostasis. And the trained muscle then is better able to meet that challenge.